Okay, so I'll continue this theme since morning of genetic diversity. And some of the work that I'm going to present, it was started with a former PhD student, Sachin Kaushik, who is now a postdoc at University of North Carolina. And we are continuing some more work with him and his current postdoc mentor, Parul Johari. Okay, so I'm again also concerned about genetic diversity within a population. And again, as you have heard many, many times, one of the big questions is simply that what evolutionary forces shape the genetic diversity in a population. So typically it's gained by mutations, and at least it's lost to genetic drift, selection, and migration and other forces perhaps. Okay, so this is a big question that you already know. And I'll be interested in the specific quantity, which I introduced to you yesterday already. That is this side frequency spectrum. So it's a sort of a slightly more involved statistics, but I hope you can get at least some idea, some understanding of it nonetheless. So let me just define it for you once, and I'll define it for you again one more time. So it's simply the mean number of segregating loci with J mutants at time t. So that's the quantity of interest for me today. Okay. So before I go to all these formulas and more descriptions, let's ask the simple question first. So we are saying that the population will get mutations, which will increase the diversity, and let's say because of drift, it will reduce it, and so on. Right? So it's possible that the population, under the action of these various forces, reaches an equilibrium after some time, and in that case, we are interested in the equilibrium diversity. However, there are other situations in which the population may reach equilibrium after some time, but there's a transient. Initially, diversity is changing with time. So in that case, if you're interested in transient times, in the initial times, at that time, the population is not in equilibrium. So that's one kind of general problems that I'm interested in. So I want to understand how diversity changes with time. Another situation is that the perhaps population never reaches equilibrium. It is always changing with time. A classical, simple, possibility is, for example, if the environment is changing with time. Suppose the population size is continually changing with time, selection is changing continually with time. Of course, the diversity will not become independent of time. So these two situations is what I will focus on later on. But I will start with the simple situation of equilibrium to begin with. And one reason I'm sort of interested in answering or looking at these problems is, well, why not? There is time. We should look at things dependent on time also. But also, for example, here, perhaps some of you already have heard of Lewontin's paradox, which says that the neutral genetic diversity is actually, in real data, is smaller than what we expect from neutral theory. So I'm not thinking only about neutral diversity, but in general diversity. It could be because of selection and other reasons as well. But one of the reasons which these authors say is that the reason we are not able to understand Lewontin's paradox is simply because population may not be in equilibrium, right? So that means it may not follow this part. Okay, so in general, let's look at diversity as a function of time. So that's what basically I'm interested in. Okay, so I will define a few things for you. I hope it's not too heavy, but if it's too much, you can come back and look at it later because it will be recorded. Okay, if you like. Okay, so I'm going to define, so I will just use the same strategy as we have been using for the last couple of days in most of the talks. That is, start thinking of a single locus, and then we will add loci to it because it's just easier to think things that way. So I spoke to you about Wright-Fisher process. Here I'm changing a little bit different. It's called a Moran process. They are similar in many ways, but let me just define this process for you because that's what I sort of find easier to work with. So then in this process, again, I'm thinking of a single locus, and we have a single mutant, which I'll denote by 1, and there are n-1 wild type. And as we were talking about fixations, just once a mutant has been introduced, we don't allow any more mutations at that locus. The fitness of the mutant, I'm denoting here by 1 plus S. S could be positive, negative, or 0, and the wild type fitness is 1. And the process is the following. It's simple. It's a birth-death process, which means an individual is chosen to give birth with a probability which is proportional to its fitness. So if the fitness is higher, it's likely to give more birth, right? That's intuitively as you expect. But then I want to keep exactly n individuals in the population. So because of birth, one individual has increased. So then we choose any individual with equal probability and remove it from the population. So that's a death. So it's a birth, one birth, and one death. That's it. Okay. And then by now, you're all experts, you know that if you just put one mutant and no more mutations are allowed, under drift, this population will reach either all fixed or all extinct, right? Okay, so great as we all know. And just to sort of say a little bit more about that, so this is a question that we had asked many, many times and seen the answers to in almost all talks. So where we ask this question that starting with a single mutant, probability that eventually we'll get all mutants. So I just want to show you that one equation here. So what we use is something called backward Fokker-Planck equation. So don't worry if you don't know it, but just for the mathematically inclined, there's nothing backward, there's nothing regressive about this equation, it's just the way it's written. It's a variation with respect to the initial condition. Okay, so it's a complicated equation whose solution is what I was showing you yesterday, which again, as you know now, so for haploids, so the fixation probability is S or 2S, I mean, I miss these factors usually, and 1 by n for neutral, and it's exponentially small probability of fixation for a deleterious mutation. So this was about fixation probability. What I'm really interested in today is about time. So let me introduce to you this concept of mean sojourn time. And it's just easiest to look in discrete space and discrete time. So let me try to explain this bit because it's a very basic thing. So suppose when the x-axis I have time, so 1, 2, 3, 4, like that, and then number of mutants are on the y-axis. So suppose we start with one mutant over here, then one is asking how many times, the average number of times it visits a site, let's say the third site. Site here just means the number of mutants before eventually it gets absorbed over here or here. How many times you visit? So John, I mean, that's a French word. So that's what it means, okay? So how many visits it makes? So for example, the blue trajectory, it visits twice. So it was here at site 3 here as well as here, right? It does not come back. So it made 2 visits. In the red trajectory, it made 1 visit here. Again, it made one visit here, right? So that's the sort of the mean number of visits that you make to a site. That is the number of mutants before eventually it gets absorbed. So that's called the mean sojourn time. That's it. Okay. So now you just keep this in your mind. But for calculation purposes, it's easier, it's better to just go to frequencies. So which means I will go to continuous space and time. Okay. So in that case, we're asking what's the mean time spent between frequency x and x plus dx, that is close to a frequency x. Then the mutant started with the frequency p before it gets eventually absorbed. We can write down a little equation for it, which says that Fokker-Planck equation that I showed you, there's a probability distribution there. So there, if you integrate it over all time, what it's saying is that it's getting absorbed at some time 0. So I want to know it appeared any time before. It started, so it's getting absorbed. So here, it's getting absorbed here at time step 10, here at 8 time step. So, but you can call that as a 0 time. And that means it started anywhere before itself, okay? So that's why I'm integrating over time. Okay, so this is called, this is how you can write down how this mean time can be written in terms of the basic Fokker-Planck equations probability. Okay, so I want to do a little bit more math. So because we know what the equation for the probability distribution is, if you plug in, the mean sojourn time obeys this equation. So if you're a physicist, if you had a MSc Masters in Physics, you know what it's called. It's called a Green's function. Okay, so it's a delta function here, and then using standard methods, one can solve this. And if you integrate over all frequencies, you get the mean absorption time. Okay, so is this all known? So don't worry too much about math, but I'll give you some simple figures here, some plots. Okay, suppose we have done all the math. So then it turns out the mean sojourn time is given by this formula, which I have just written in a simple manner like this. Let me explain that bit to you. So if selection is positive, then the mean sojourn time is a U-shaped function. Okay, green is beneficial, neutral is blue, and red is deleterious all through. So it's a U-shaped function. If it's a positively selected mutant, it's decreasing function, which is I showed you yesterday when we were discussing yesterday morning. If it's a deleterious mutation, again, it's a decreasing function, but it decreases much faster than the neutral case. And that is intuitively clear because, like we said, discussed yesterday, the neutral case, because we're starting with a single mutant, most of the time it will hang around 0, 1, I mean like 1, 2, 3, and will get absorbed. So it's unlikely that it will reach high frequencies, right? And therefore, it will be, the chances of getting high frequencies will be low, and that's why it's a decreasing function. If it's a deleterious mutation, it's the chances are even worse, and that's why it decreases even faster. But for a beneficial mutant, it has a high chance of getting up to the high frequency, right? So therefore, it initially decreases and then it increases. So that's a U-shaped function. So I want you to just keep these three very qualitatively different looking plots in mind. So that's the basic thing that I want you to remember from this talk. Okay, so that was just a single locus, and I spoke to you about sojourn time. So what has that to do with site frequency spectrum? So let's go to that direction now. So just to recall again, so unfolded site frequency spectrum, unfolded simply means I'm assuming that if I have a data of this sort, these are my 6 samples, and then these are lots of loci, I want to know how many loci are polymorphic and they have J number of mutants. And we did this little histogram yesterday. Okay, so how do we model this? So this is from the 90s, people have thought about it, and there is this very standard sort of model which has been introduced and studied quite a lot. So by Sawyer and Hartle. Okay, so the model is based on the following idea: that suppose you have lots and lots of loci, that's an infinite site model. Okay, and suppose there is free recombination, that means a lot of recombination between different loci, so that I will just ignore LD completely. Secondly, because there are lots of loci, once a mutation has occurred, I turn off the mutation there simply because the probability of hitting the same site again is negligible. So that's the infinite sites model of Kimura. But now let's just look at these trajectories in this. So what happens? So at the first locus, a mutant is present, one mutant, and then it does its thing, maybe it increases a little bit, and then we expect it will probably go to extinction. Then at another later time, stochastically, there is some clock which says, yeah, produce a mutant. And that produced a mutant, which is the second locus. Third one, another locus, and maybe that reached high frequencies and so on. Okay, so these various trajectories represent various loci at which mutants are appearing. And then once a mutant has appeared, it's evolving under the action of drift, selection, and perhaps other processes. Okay, so just to again finish the point about equilibrium SFS, trajectories are being lost due to absorption and maybe getting created due to new mutations. And therefore, as we discussed many, many times by now, that a mutation drift balance or mutation drift selection balance can be reached. So in that case, you can ask, what does the SFS look like? So that SFS is actually connected to the mean sojourn time of the Moran process that I described to you. So again, it may need you some to think, so go home and think about it, but I'll give you the answer. What it's saying is that at long times, when I'm looking through these trajectories, okay, so it's like if I'm looking at this time slice, it has reached some frequency. So it will get contribution from all the things that came before it. So that's this integral I'm writing here, okay? And that was proportional to this Green's function or the sojourn time I spoke to you before, okay? So the key point I want to make here is simply that this multi-locus problem, by virtue of being a single locus problem related to that, we can use those results, and simply SFS here is proportional to the single locus answer. So that's not surprising. Okay, so what is it good for? What is it? Why should we bother? So because if we assume there's equilibrium, then this SFS is used to infer if there's selection. So for instance, if you see this U-shaped function, then it suggests that S is positive, that is selection is a positive selection. If it is there, decaying, negative, and so on, and then you can do certain likelihood analysis and other things to figure out various parameters of your model. Okay, so this is the segregating loci. Also, you can find out using SFS. Okay, so that was all the sort of the introduction I wanted to give. But what I'm really interested in is the diversity in non-equilibrium populations. So we can look at dynamics of SFS in constant environments. That means population size is constant in time, selection coefficient is constant time, and maybe other things are also constant in time. So there has been some amount of interest and some results have been obtained. I think the first paper on this is from Steve Evans' group. He's a probabilist. His model, which I will describe, and then all kinds of complicated situations, including, for example, polygenic selection. So people have started to look at the SFS or other diversity measures as a function of time, rather, for the last 2 decades or so. You can also look at SFS, you can at least ask about SFS in time in homogeneous environments. So a classical thing which has been done for a long time is changing population size. So people have looked at heterozygosity, for example, or other measures of diversity, but not SFS when population size is changing. Okay, and that, as far as I know, is the first paper, I think, for SFS is over here. Then with changing selection alone is also very less has been done. And then you can ask, what if population size and selection, both of them change? So as far as I know, hardly anything has been done. Okay, so this is what I'm going to use for my analytical model. So I said to you that I'm interested in this SFS. It can be related to the results of the diffusion theory. Now I'm interested in a situation where population size is changing also with time. So 2n mu, which was the rate at which mutations were appearing. So now that is also under the integral sign. Okay, so that's the sort of simple thing which happens. Where now, for this, I'm going to use the forward Fokker-Planck equation. And this is the model of Steve Evans, Schwartz, and Slatkin, in which this equation is same as the usual Fokker-Planck equation, the forward one, when you write down for Moran process, except here, these coefficients are time dependent. But this is selection and drift, and they model the mutational input because they'll get absorbed. So therefore, you need to have constant mutational input. So they model it using a boundary condition. So that's the model that we are using here. Okay, and the model, let me just explain this to you. So it's saying that when the frequency is very, very small, no forces are important except genetic drift. So there, the SFS is simply the answer I said earlier, which is 2n mu by x. So they model that this mutational input through this boundary condition, and this is the other boundary. Okay. Okay, so we have this complicated differential equation, we have this boundary condition, it's very, very complex, and so on and so forth. And it's impossible to basically obtain exact solutions, except for one case, which is when selection is not there. And that is simply, and that's very well known because of the work of Kimura. Okay, so we are interested in, so I'll give it a couple of examples from what we are interested in. So I want to describe these 2 problems. So let's take this first case in which I'm assuming that both selection and population size is changing, and both are changing in a periodic fashion. So for example, because of seasonal variations, selection may be changing, and demography because of, for example, predatory dynamics or some other reason. Okay, so let's go back to our infinite loci model of Moran process, which I described to you, and we will look in again this our Fokker-Planck equation. So we can define this very general model, which has lots of parameters. There's a mean, there's amplitude, different frequencies. There could be phase difference between selection and population size. So there's a lot of parameters, and we can make some analytical progress. But today I will describe basically only this case, which is when the environment is changing very slowly. And we can look then at how the mean F averaged over one period of time changes. What is that value? Okay. So let me just describe this little idea because it's a simple intuitive idea. So if the environment is changing very slowly, more precisely, environment is changing slowly means the time over which environment is changing is longer than the time on which selection acts and the drift acts. So that's the sort of limiting case. So now there's an intuitive idea, which is in physics literature, at least, it's called adiabatic approximation. So what it's saying is that your environment has changed a little, but it's so little, it's changed slowly so that your population comes to an equilibrium. So things have changed, but population quickly equilibrates, but to a new value. So now you calculate the SFS when your S selection has changed, plug in the new instantaneous parameters in the equilibrium answer. So it's a very simple idea, and we know that it works. Okay, so that's the approximation that we will use simply because, as I said, we cannot solve Fokker-Planck equation otherwise. Okay, so we can do, so if we do that, then in the absence of selection, we have the SFS time dependent SFS was 2 times 2n mu by x, so n was time dependent. If you average it, then it's linear in n, nothing happens, and it just looks like it's just seeing the mean population size. But then when there's selection, but because there's a huge asymmetry in how positive selection and negative selection works, because of that, the SFS when selection is positive has this expression. Okay, so it is this 1 by, which is this parabola part. For negative selection, it is this decreasing function. But now if I average over a period, again, you know this, for strong selection, you do not expect any contribution, I mean, negligible contribution, right? Because it's, if the selection is strongly negative. So it's just this little simple thing. So if I now average over a period, this full f of xt, it gives me this result. So let me just explain what it means, what implication it has. Okay. Okay, so this is the SFS at times when my population size and selection both are changing periodically, let's say right now with the same frequency. And I have averaged it over a period. And what it's saying is that the final answer has x dependencies like this U-shaped. But this part is saying that you will get a contribution from the population size only where selection is positive. So this is called theta function, okay? So it's just saying selection wherever is positive, that's where the contribution will come from. So the consequences are, we think, are the following, that even if selection was 0 on an average, so half the time selection is positive, half the time is negative, right? There's no contribution coming from the negative selection part, only from the positive selection part. So therefore, although there is selection, it's neutral on an average, okay? Yet your SFS will look like, so if there is neutral on an average, perhaps you would think that SFS on an average should look like the blue curve. But because it's not getting contribution from the negative cycle, it looks like a U-shaped function. Okay, it's a simple thing. Okay, it's just getting contribution from the positive part, no negative part is contributing, and that's why it looks like a U-shaped function. Although perhaps you would expect, because if it's neutral on an average, it should look like a decreasing function. Secondly, it looks like the answer for the positive selection into some slightly complicated looking object, which we call an effective. Okay, so it looks like it's positive selection, but there's a new effective population size. Okay, so that's our effective population size, and one can look at it whether it's small, large, and so on. But I will not go into that. You can look at the other extreme, which is a rapidly varying environment, and this result is known for a long time. That if there's no selection, in that case, but however population size is changing with time, then again it is known for a long time in the literature that the time average SFS is 1 by x, the usual answer, times the effective population size, which is simply the harmonic mean. But as far as I know, it was checked only in simulations. I don't know if there's any proof, but we are able to show this. The selection, we tried a few things, but it did not quite work out. Few things that we tried, it was not working very well. So I don't have a good understanding of that part in the fast changing environments. Okay. Okay, so I want to now describe one more thing quickly. So you heard about selective sweeps already in the morning. So again, we are considering Moran process, which I'm just considering 2 loci. And unlike what Mike said in the morning about with recombination, I'm interested in selective sweeps in asexuals. So first, let's consider 2 loci, and they're fully linked for always. So the first locus is neutral, which I'll denote by alleles there by 0 and 1. Other one, the first locus, second locus is neutral, and first locus is selected. And here I'm denoting allele by dash or star. The dash has a fitness 1, and star has a fitness 1 plus s. S is positive. So due to selection of the selected site, if provided a star does not get lost, then what would happen very deterministically? So this is time, and these are the individuals. So let's say the star mutation came here, And then because, if it does not get lost, it will spread in the population, and all the population becomes star 1, star 1, star 1. Initially, you had half population which had 0 and half population had 1. So you had neutral variation in the beginning, but now you've completely lost it, right? So that's the standard thing that we'd already heard in the morning. But now we wanted to ask that what if, so we don't want to put recombination. We want to look at this for asexuals. So suppose we look at this, not 2 loci, but many, many loci. And now we have neutral mutations raining down on each locus. Not raining down, like it just comes one mutation only irreversibly at each locus. So again, it's the old Kimura's model, infinite loci with irreversible mutation at each new locus. But now loci are fully linked. This is an asexual population. So we are generating, therefore, diversity at neutral loci, although it's getting lost also because of drift and selection. So I'm just drawing things here differently. So 0 time, I have this dash 0, dash 0, dash 0. And at this locus, selection happens. And because it is selected, therefore, it produces more offspring here. And then maybe second locus, at second locus, neutral mutation occurs, but that must appear at a new site, right? So therefore, at the second locus, I have here a 0 and a 1 here, right? Because I have to add it at always a new site. And then if I just repeat this process, I find that still we have diversity left. Okay, so it's different from the usual models of asexual populations where you don't say that new neutral mutations are also coming. So there's no recombination here, but diversity is there because loci have neutral mutations. Okay, so we just wanted to understand that. Okay, so how do we go about it? Yeah, question? No, okay. Yeah, so we're interested in the diversity, especially at the end of the sweep, that how much it is. And since we are interested in this star subpopulation, the ones that has selected site, so this is very difficult. Asexual population fully linked is just almost impossible to do anything much. But here is what we are doing. The full model has selection, and it also has a fixed population size n. But if I'm interested in population with the star only, then I removed all the other stuff and I just track the population with the star, that is the one which is selected site. So in that case, it's a population of growing size. It will grow, right? Because it's selected one. But all these 1 zeros and 0 ones only are there. So it's fully neutral within, but in an expanding population. So we are reducing the problem to this sort of problem. Okay, except we cannot make progress with that either because they are still selected. Still linked. So if I assume now that, yeah, all these neutral loci are also still independent to me because otherwise I simply cannot do anything. So we still have, however, a expanding population, and we change the Moran process a little bit in which the population may remain same, it can increase, decrease, and we can write down what the probabilities are for that. And then try to write down the new diffusion theory for that. This one has to be a little bit careful whenever population size is changing, because when we write down for frequency, only we say the allele number has changed, population size, the denominator is constant. But now allele number is changing, as well as population size is also changing. Because of that, some new terms come into play. Okay, so we do that, and this is the usual neutral equation we get. But this effective population size is not the naive answer. It's a new one, which I did not write the formula though. But again, some effective population size, which is changing with time, is what we can define. The P was the frequency, which we saw already in the morning. Okay, does it work? Because I made this important assumption for the asexuals that the loci are linked. Simulations seem to indicate that it works pretty well. It's not, it's not very off. I think we have better data, which I did not replace, but it seems to work well. Also, the answers are different from the standard neutral answer. That is, it's not, the neutral answer is 1 by x, but for small frequencies, 1 by x, and then it becomes 1 by x squared. This 1 by x squared is not really a new result. It has been seen in the context of cancer, and I think Duret had derived it first. But there you just assume that it's an expanding population. But as far as I know, in the sweep literature, that kind of effective population size starting from the full model has not been derived. Okay? Okay, but this result is sort of known in some ways. So what we are actually right now doing, we have some results which I have not shown you, is the how SFS changes with time. So that I think we're just fixing some factors right now. So okay, so I think that's all I wanted to say. So I just interested in understanding how diversity, SFS, etc., various statistics, how do they change with time, not just at the end of everything is done and balance has reached. So those are the questions that I wanted to understand. And two situations I considered where we never have an equilibrium, for example, in time inhomogeneous environments, or the system is in transient state, for example, the case when the population is expanding, but it reaches to some maximum. So there are some things which we did not at least expect, but yeah. Okay, I'll stop here. Thank you very much. Okay, yeah.
Thanks, very exciting. I have just one question to understand like the equation you gave for how SFS are proportional to the beyond time.
Yeah, because roughly, because each locus is independent. So each locus is independent, it's getting its mutation at different time. But if you look at large times, it doesn't matter when it came. I mean, you look at all the times it came in. So it's basically because it's one locus. I mean, it's unlinked loci. It's roughly that. Yeah. Yeah. No. You start from the Moran process, which is a discrete, yeah. And then you write down for mean and variance and you plug it in. So it's derived from the Moran process, what you assume, and then you work it out. So that's standard. I mean, it's not special. Okay, sure. Okay, so it's slightly different from the usual process. I mean, it's just, yeah, I'll say it once more. So again, once more. Oh, I see. Oh, okay, sorry. Yeah, so this is the birth death part in which population size remains constant. But I'm saying now, if the size of the population increased by 1, because we allow one death, one additional death or additional birth, then you just add one individual. It could be 0 or a 1, equal probability. Similarly, you just take out one, either 0 or 1 with equal probability. That's all. Yeah. Okay. Okay, fine. Let's stop here. Thank you very much.